very happy to introduce Kimberly McDowell from Capsida Biotherapeutics, and she'll be speaking on systemic AAV gene therapy with CNS-targeted engineered capsids, which achieves significant GCase activity increases this is the longest title, GCase activity increases in primate brain to support the potential treatment of PDGBA1. Thank you.
Thank you. I was aiming for longest title of the morning. No, thank you. It's really a pleasure to be able to speak with all of you today. Make sure I do this correctly. My disclosure is I work for Capsida. So I wanted to first go into a little bit of introduction of what Capsida does. So we're based in Thousand Oaks, California. And really we were founded on technology based on AAV capsid engineering. So really to design off of AAV wild-type serotypes that can create a new class of targeted gene therapies that can be delivered systemically, so non-invasively, to really cross that blood-brain barrier. And so as part of the work that we've been doing, we have a variety of partnered programs, but we also have a pipeline that consists of internal programs, one of them being PDGBA, which is the one I'll talk about today, and the one I'm very happy to lead at Capsida. We're also a fully integrated company, which is very handy in the sense that we start with capsid engineering and cargo optimization. Once we find certain leads, then we go into discovery and preclinical research, but then have the capability as we reach IND enabling stage to then also look at manufacturing and begin clinical development. So again, as I mentioned, our company was founded on trying to address some of the challenges with being able to treat CNS-based diseases systemically with AAV-based gene therapies. So when you deliver something like a wild-type AAV9 systemically or intravenously, it's going to have a limitation in being able to cross the blood-brain barrier. And so the example, if I can get the mouse, the example shown here is in a non-human primate cortex. And so you can see there's a little bit of a counterstain there with immunohistochemistry, but you don't really see robust transduction of neurons, again, when AAV9 is administered IV. Also, as part of that challenge, then you might want to try to increase the dose to get more of that wild-type AAV across the blood-brain barrier. But again, that poses other concerns with safety. So things like liver or dorsal root ganglion toxicity. And so then basically ultimately what you land with is potentially a narrow therapeutic index in which you can treat CNS disorders. There are ways around that. And so again, you can do direct injections, either directly into the brain or through intra-CSF injections. One thing with direct injections into the brain is you're going to have a limit in how widespread the transduction is going to be across the brain. So if you really want to hit a lot of areas of the brain, the direct injection has some limitations. So what Capsida does is we have directed evolution and an engineering platform where we screen billions of variants, basically, in non-human primates. Because again, it's important to keep an eye on translatability, especially with designing novel AAV capsids. So as you can see, again, in the, this histological image, so in this case, we have an HA-tagged cargo that we assessed looking with immunohistochemistry and can see a robust increase in the number of neurons that are stained in the cortex of a non-human primate. So again, we can cross the blood-brain barrier after a single intravenous administration in NHPs and hit up to 70% of neurons throughout the brain. What we've also done as part of this engineering is basically modify the ratio of how much of the virus is getting into the brain across that blood-brain barrier and how much of it is getting in peripheral organs like the liver. Again, that's designed not to completely de-target the liver, but to, again, reduce chances for safety concerns or other immune responses that you can get with AAV administration systemically. So it's a little bit of a complex slide, but what this is highlighting is some of the lead capsids that we've identified at what we call a single variant level, meaning instead of looking at those billions of capsids, we're looking at just one individually in each dosed NHP. And so here we're on the graph on the left, if I'm not confusing everything, we can see the biodistribution at both a DNA and an RNA level. So with this graph, what's set at 1 is this is normalized to wild type AAV9 that would be delivered systemically. And this is log scale. So above wild type AAV9, you're looking at different brain regions. And again, what that is highlighting is with our different capsids, we are seeing robust transduction and higher biodistribution than wild type levels. And what you'll see at the very start of the graph that's lower than AAV9 is that we are detargeting the liver. And then the last piece here is that, again, we have some histochemical images highlighting after one of our lead capsids, again, were injected. You can see robust staining in the cortex of the NHPs with reduced expression in the liver. So a little bit before we get into specifically what we call CAP003, which is our PD GBA program, we do also have other wholly owned programs and also some partnered programs. So I just wanted to acknowledge that. So this slide, really, I'm not going to cover because you guys are all the experts in this. And so what I will highlight, though, is, again, specifically with the candidate that we identified for our PDGBA program, is that we are able to transduce very robustly in NHPs after systemic administration of our drug product. And again, that's over 50% in the substantia nigra, so we know that's a critical region. I'll also show some other brain regions as well in a minute. And importantly, especially for this group, we also looked at GCase activity levels and do see elevation. So again, this is in wild type or normal NHPs, but we are able to look at the expression, increase in expression and activity over those wild type levels. So before we jump into that non-human primate data, so this is data that we used using, and I am going to use the term loss of function. In a GBA1 loss of function model, so this is a homozygous mutation where you can see, so if we start just by looking at what the genotype difference is you can see at a GCase protein level, you're not seeing a reduction in protein, but what you are seeing is about a 90% reduction in GCase activity. And again, that reduction in activity in the brain, so all of this is brain-based data, you do see an increase in the glucosal sphingosine levels, a marked increase. It's basically undetectable in wild-type mice. So what we see when we administer our therapeutic cargo, so we'll give back the GBA1 gene, so we are producing protein in a dose-dependent manner, but importantly, that protein is also causing an increase in activity in the brain, and that increase in activity is leading to a reduction in the sphingolipids, so confirming that we do have target engagement with our therapeutic cargo. So now into one of my favorite slides because I always love the pretty pictures. So in this case, we're doing immunofluorescence. So what's in magenta is, in this particular study, we used an HA tag, again, on that GBA1 cargo. And so what you see is a variety of different brain regions. For the substantia nigra, the counter stain is tyrosine hydroxylase, so we're looking at dopaminergic neurons. In the other brain regions, like thalamus, cortex, and putamen, we're using NUN as a neuronal marker. And so when we go through and actually quantify the levels of transduction that we're seeing across the brain, again, we're seeing that we're hitting not just cortical regions, but also subcortical regions, and importantly, hitting the substantia nigra at over 50%. So one of the things that has been great to hear about over the past couple days are the differences in GCase activity assays, different probes that might be available, substrates, inhibitors. So when we started off some of our early work, we used sort of a very common in the literature research grade assay, and to look at increased GCase activity levels. So just to orient you again, these are normal, healthy NHPs. So there will be endogenous and normal levels of GCase activity. So what we're reporting here is the percent increase over wild type. So we can see in multiple key brain regions of interest, so whether that be the substantia nigra, caudate, putamen, frontal cortex, or thalamus, we are seeing notable increases, again, after we've administered, this is, in our case, 6 weeks after we administered that single IV injection. And so, again, based on kind of literature, we've also heard a lot about how the disease can be very heterogeneous. But again, somewhere between 25, 30, maybe in some cases 50% decreased in average brain, decreased GCase activity on average in the brain, looking at some available published post-mortem data. So again, these increases looked promising in terms of being able to normalize enzyme activity defects. But so again, with this assay, we wanted to take it one step further. So as we are gearing up, we are in IND enabling stage right now. So we wanted to switch to a different clinically validated GCase activity assay. Again, this is based on mass spectrometry and uses more of a GCase1 specific substrate. So again, the talk earlier this morning was really great to see in terms of the specificity of different options. So again, what's shown on the left is just an estimate. Again, this is adapted from a published paper, is that you are seeing maybe around 30% decrease in the average activity in the brain. Again, that was using, in that particular paper was using the 4-mug assay with the subtractive CBE method. So we learned that there might be some variability there. But again, so moving to this other method, we see using the frontal cortex and putamen as examples, that when we are using a GCase1 specific substrate and a mass spec method, we are actually detecting even higher levels of increases in GCase activity. And again, that may be due because the previous assay might have had some GBA2 compounds in it. But importantly, again, these levels we do expect to provide clinically meaningful benefit. The last thing I'll say before I wrap up is, so one of the things I mentioned at the start of the conversation was that we do detarget from the liver. That does help because, again, we are doing systemic administration, so we want to make sure that we're not posing any safety concerns. So in this case with the CAP03 drug product, it is about 16-fold detargeted in the liver compared to AAV9. And so again, AAV9 has, when delivered systemically, does have robust tropism in the liver. So again, overall, during the study that I showed you data from in those NHPs, again, it was very well tolerated at all the doses tested. Didn't see significant or remarkable clinical pathology, immune responses, or histopathology, which is promising. So with that, I'll wrap up in that, again, the candidate that we've selected for our PDGBA program is able to efficiently cross the blood-brain barrier and really be able to show robust and widespread expression in the brain. That is also goes along with increase in GCase activity over those wild-type endogenous levels. And again, in therapeutically relevant areas of that NHP brain. And again, this efficacy was achieved. The data that I was showing you is from a relatively low dose that is expected to potentially be able to achieve therapeutic or clinically meaningful levels without raising safety concerns. And again, as I mentioned, we're a fully integrated company. So again, our manufacturing team is amazing, and they have already been working and confirmed that the CAP03 is able to scale well, and that we will be able to meet sufficient quality or quantity needs to be able to progress into the clinic. And one other thing that I mentioned again is the importance of translatability. So we have characterized the human receptor that does bind the engineered capsids, and we did assess the homology between the macaques and the humans and do find completely conserved homology, which is, again, very important for translatability. And so with that, I'll just say that I am really excited that the program, again, we're in IND enabling stage, so we're quite busy, but it's for a really exciting goal, which again is starting clinical trials next year. So thank you.
Thank you. That was great. So one quick question, we've been told.
Always quick question. With which mouse model did you use, and did you show any phenotypic improvement?
So now I don't know which one I'm supposed to use. Sorry. Either. Okay, great. So we used, it was a homozygous GBA1 model. And we didn't look at phenotypic correction. It was a short-term study.
So do you
For
have
proof of concept.
any evidence in any kind of Gaucher model that it's going to make any impact on the disease?
So that was what we used for proof of concept, which is more of a Gaucher's model because of the very strong reduction in GCase activity. Current ongoing studies are looking at other models that mimic.
What point mutation was it? Which model was it?
We can maybe chat after the call.
Okay. And as far as the design, what kind of promoter and what kind of construct do you put together? Is that all proprietary?
That's proprietary, yeah.
All right, so I want to thank the speakers again for some wonderful talks, and thank you all for your attention, and especially to Ziv and the organizers.